Hi, I'm Murad, and the person behind the camera is BIOL. So, this is our honors project, and today we're going to provide you all the information you need about zebrafish. First, we're going to delve into some background information, but then our main focus would be how to maintain zebrafish for your labs. So, why do we use zebrafish? We use zebrafish because they are what you would call a model species. This means that they can be used to study systems in other organisms. They have a body system that is very similar to that of us humans, and they can be used to study the effects of alcohol, toxins, and other drugs on blood circulation or the immune system, for example. They have translucent skin, which means it is very easy for us to see the effects of drugs on their blood circulation or their heartbeat, for example, and they reproduce and grow rapidly. They're cheaper and easier to maintain compared to other alternative species, model species such as rats or mice. And all of this means that they can be utilized for the purposes of understanding systems in other species.
Before you can do anything, you need to put on gloves, just like these, whatever size it fits. And depending on what chemicals you're working with, you may need to put on goggles, just like these. After you put on your gloves and goggles, if you need them, the first big step that you need to do when you're cleaning out your dish is you need to go and visually inspect your dish to see what kind of contaminants or other materials like chorions or dead zebrafish, which we will get to in the next video. See what is in them, and preferably you should do this with using the microscope, but sometimes you can just visually inspect it. Here we are showing you a worst case scenario where someone spilled contaminants into your dish, and now you have a whole lot of debris and other things that you need to clean up. And you can see the debris, these black little spots, all around your dish. You also see, these are the embryos right here, and this is a dead embryo that we will need to remove. The chorion surrounds each embryo, and oftentimes you will find the chorion floating around, and you will need to remove that too. In our case, you can see we have lots of things to remove. So let me show you. So how you do it is you take your pipette, preferably 1,000 microliters, and you just look and you suck up as many contaminants as you can in one go. We just got done cleaning out all the dirt and debris that we had in there, and now what we have remaining are dead zebrafish embryos and alive ones. And how do you tell the difference? Usually for embryos, they will either look white, they can either look like there's a speck of white inside of them, they also could look blackish gray. In our case, we have primarily white looking dead embryos. You can see here spread throughout our dish, white ones. If they are adult embryos though, you usually can tell if they're dead if they are either cloudy or just they look gross or deformed. You can double check if you're not sure by just looking for their heart rate, and you can ask your TA how to do that. But in our case, you take your micro pipette. You want to make sure that you have the tip cut. You want to cut off the tip so that you have a wider opening so that the embryo will fit into the pipette. And then you carefully suck up each dead embryo that you see. And you continue to do that until you have all the dead embryos. And once you're done with them, carry them over to something we call the embryo morgue. So once you have to open it up, which I can't open up in this case. But all you gotta do is put it in there and you're done. After you identify and you suck up all the dead embryos that you can find in this micropipette, as long as your embryos are younger than 7 days old, you can dispose of them into the morgue just by doing this. However, you have to be careful because if they are 8 days or older, then you have to follow a different method where you put them into a solution and you put them into the freezer so that they basically freeze to death. And that goes for all embryos older than 8. There are resources online listed under your class that is for this if you want to look into it, but that's about all you need to know.
Yeah, so now that we have removed the dead embryos from the petri dish, what we want to do next is replace the embryo water. So we're going to get down and eyeball the petri dish, and we want to replace at least 10% of the embryo water in there. A good rule of thumb is to replace more than less. Now that we have sucked up some embryo water, we're going to dump that into the liquid waste beaker, and we're going to follow that up by adding embryo water, which is somewhat equivalent to the amount that we have removed. So after you're done with cleaning, you need to ensure that you label your petri dish. This is to make sure that others know that you have cleaned up the petri dish so that they don't end up re-cleaning it. So here's what you do. Just take this tape and rip off a piece of it, and yeah, put that on top of the petri dish, and you write clean on the date, and that's good enough. So one important aspect of keeping zebrafish alive is feeding them. You wouldn't have to do that for zebrafish that are under 6 days because they have that amazing little thing called the egg yolk, which sustains them. And for zebrafish that are older than 6 days, you would have to provide them some alternate nutrition. This could be in the form of rotifers, which could be provided to you by a TA.